Welcome to the second part of our lecture on the introduction to protozoa. In this presentation, we are going to discuss the encystment of the protozoa and its importance. We are also going to discuss the different economic importance of the protozoa, the harmful damage that is caused by the protozoa that is also known as its pathogenesis, as well as its mode of transmission. So how do we define encystment? Encystment is said to be a feature of many protozoan life cycles, especially those living in environments that periodically dry out. So it is said that many of the protozoa form a resistant, dormant structure called a cyst. Many protozoa also produce cysts under adverse environmental conditions such as food deprivation. The diagram shows the cyst form and the active form of the Giardia lamblia. So this is the cyst form of the Giardia lamblia. This diagram shows the cyst and the trophozoite of the Entamoeba histolytica. So the trophozoite form of the parasite is said to be its active stage, while the cyst is its inactive or dormant stage. So the trophozoite in this diagram is characterized by the presence of the nucleus as well as the engulfed red blood cells. The cyst, on the other hand, is characterized by the presence of the hard outer covering as well as a single nucleus. When this is mature, it contains 4 nuclei. So what are the functions of the cyst? The cyst endows protection against unfavorable environment, as exemplified by Entamoeba histolytica. Another is nuclear division can be followed by multiplication at the time of encystment. This is also exemplified again by E. histolytica. Third, the cyst may also function in attachment, as in the ciliates of certain fish, and it also serves as a means of transmission from one end to another. Example is in the case of Entamoeba histolytica and Giardia intestinalis. The diagram shows the cyst of the Entamoeba histolytica. So this is a cyst, mature cyst of the Entamoeba histolytica containing 4 nuclei. We also have here the trophozoite form of the Entamoeba histolytica, showing the vesicular nucleus as well as the presence of the red blood cells. There are different factors that lead to the formation of the cyst. First, cysts can form when there is nutrient deficiency, desiccation, increase in the concentration of dissolved salts in the medium, changes in temperature, low pH, decreased oxygen supply, accumulation of waste products in the medium, as well as the crowding phenomenon. This table shows the major differences between two pathogenic organisms, the protozoa and the bacteria. The protozoa is considered to be eukaryotic, while the bacteria are prokaryotic cells. Protozoa have a distinct nucleus, while the bacteria have no distinct nucleus, so that its nuclear material are dispersed. Protozoa have a distinct double-layer nuclear membrane, while the bacteria has no nuclear membrane to separate the nuclear apparatuses from the surrounding cell. For protozoa, they have a highly developed and organized organelles such as the ER, the Golgi, the mitochondria, and the lysosome, whereas the bacteria has the absence of these features. For protozoa, the infection that is caused by protozoa runs a protracted or a prolonged or extended course, while for bacteria, the bacterial disease progresses at a first speed. Let's now proceed to the economic importance of the protozoa. So it is said that majority of the protozoa are free-living, of which the large-sized protozoa play an important role in the control of bacteria in water having dead organic matter. Thus, they play a vital role in sanitary microbiology. In addition, The ciliates are normally found in the stomach of ruminants and in the cecum and the colon of equines, and they form an essential part of an environment in which the metabolic activities of the host, the bacteria, and the protozoa are all linked. Another major economic importance of parasitic protozoa is that it can cause disease in domestic animals and human beings. Some of the protozoa disease appears with severity, resulting in mortality. So the protozoan disease can present as either acute, subacute, or chronic form. Both the subacute and the chronic form may lead to poor growth, loss of body weight, as well as reduced milk, egg, and wool production. Let's now proceed to the harmful nature or the pathogenesis of the protozoan infection. Protozoa are too small to cause mechanical obstruction. However, occasional clumping of the organisms may lead to blocking of the capillaries of the visceral organs, including the brain and the spleen, and this can be exemplified by the hemoflagellates such as the Plasmodium, Babesia species, and the Trypanosomes. So the protozoa, particularly those protozoa that resides in the blood, they are very important because they can cause mechanical obstruction of the blood vessels. Protozoal infection can cause the development of hematological changes in the host. All blood protozoa cause a variety of changes in the normal blood picture. For example, anemia due to the destruction of the RBC and des hemopoiesis can be exemplified by the infection with Trypanosoma congolense. Another is the hypoglycemia. So hypoglycemia can develop due to the consumption of a large amount of blood sugar by the parasite Trypanosoma evansi. We also have the exhaustion of the glycogen reserve in the body due to liver dysfunction, increase in lactic acid, which interfere with respiration. For pyroplasms, such as the Teleria and the Babesia, they can cause hemolytic anemia because they have the ability to destroy the RBC. We also have the development of leukopenia arising due to the prevention of the normal development of the granulocytes. And this can be demonstrated by the parasite Leishmania donovani. This diagram shows the histological section of a brain tissue of a dog that developed severe clinical signs upon infection with Babesia canis infected erythrocytes. So when we say Babesia canis, now that is an example of a blood parasite and it is a pyroplasm under the phylum Apicomplexa. So in this diagram, the capillary appears entirely blocked and surrounded with perivascular edema. The insert shows a part of the capillary with high numbers of the parasites. So the parasites, Babesia canis, have resulted to the blockage of the capillary. Protozoa infection can also cause the development of jaundice. This is due to the occlusion of the bile ducts caused by parenchymatous degeneration in pyroplasmosis. So when we say pyroplasmosis, that relates to the infection of the blood parasites, Babesia and Teleria. There can also be fall in the RBC count, paxil volume, and hemoglobin, increase in the white blood cells in Teleriosis. This diagram shows the post-mortem findings of acute babesiosis in an 11-day-old calf. For letter A, we have here the urinary bladder with hemoglobinuria and also jaundice in the liver. This is an enlarged spleen, enlarged liver, and enlarged gallbladder, and of course, we have here a picture of a liver with jaundice. Another consequence of protozoa infection is that it can interfere with food digestion. Giardia lamblia is responsible for mechanical interference of fat absorption from the intestine by the layer of parasites adhering to its wall, which leads to a deficiency of the fat-soluble vitamins. In the case of coccidia infection or coccidiosis, coccidial recovery results in replacement of the functional epithelium by connective tissue, which hamper the intestinal digestion and keeps the individual unthrifty. Protozoal infection can cause interference with food digestion. This can be exemplified by trichomonas infection or trichomoniasis. Trichomoniasis of the upper digestive tract result in yellow caseous lesions on the oral mucosa, which extend to the esophagus, crop, and even trachea. As a result, the bird will avoid feeding due to painful lesions. This diagram shows the oral cavity of a pigeon with lesions consistent with trichomoniasis, including the presence of the caseous necrosis. So the presence of the caseous necrosis in the upper digestive tract can cause interference with food digestion because it hampers the feeding habit of the bird. Protozoal infection can also cause the destruction of the host cell. All intracellular protozoa are responsible for the destruction of a large number of cells and various organs and tissues of the host. For example, the hemoprotozoan can cause the destruction of the RBC. Destruction of the GIT epithelial cells can be exemplified by the coccidian species such as Eimeria. Coccidia can cause the rupture of the cells and the blood vessels. For the malarial parasites such as the Plasmodium, they can cause the rupture of the red blood cells. For Entamoeba histolytica, they can use the red blood cell as a source of food. This diagram shows the schematic figure of the intraerythrocytic cycle of the Plasmodium falciparum. Plasmodium falciparum is the causative agent of malaria. The intraerythrocytic cycle of the parasite is one of the stages in its life cycle. When we say intraerythrocytic, that means the development of the parasite within the erythrocyte. So within this diagram shows the merozoites. Now the merozoites comes from the liver stages of the parasite. The merozoites are released in the bloodstream and it will later invade the erythrocyte. Then within the erythrocyte, the parasite will later develop into its ring stage. After the ring stage, it will become a trophozoite, in the trophozoite stage, and the trophozoite will undergo schizogony. So during the schizon stage, so the nuclei will divide several times mitotically, and it will later develop into new merozoites. The mature merozoites will rupture the RBC and will be released in the bloodstream, ready to invade new cells and complete the cycle. So in this diagram, we can see that the parasite Plasmodium falciparum causes the destruction of the erythrocyte. Parasitic protozoa may also cause various host reactions. These include the development of inflammation, hypertrophy, hyperplasia, increase in the peristalsis as demonstrated when there is an infection with Entamoeba histolytica, in amoebiasis, enlarged lymph glands, as in theileriosis. There can also be swelling of the spleen and the liver, and hyperemia in the kidney as demonstrated in the infection with trypanosomes. In the case of the plasmodium, so it can cause the development of splenomegaly or the enlargement of the spleen due to its exoerythrocytic infection. In the case of Eimeriaceae, so it can cause tumor-like lesions in the bile duct. Protozoa infection can also lead to the production of toxins. There can be necrosis and lysis of the tissue that may be due to various toxic substances and proteolytic enzymes having cytolytic effect. This can be caused by the presence of Entamoeba histolytica and Balantidium coli infection. Trypanosomes elaborate toxins, which result in the production of antibodies, the ablastines. We also have the Plasmodium. So Plasmodium species, in addition to destroying directly the RBC, it may also cause the hemolysis of the uninfected red blood cells. For the mode of transmissions of the protozoan parasites, so this can be divided into two. We have the direct and the indirect mode of transmission. So for the direct mode, we have the ingestion of the contaminated feeds and drink. So the feed in the drink can be contaminated with oocyst. The cyst of the Giardia, Entamoeba, Balantidium coli, and others. There can also be ingestion of the infected meat containing stages of the parasite, as in Trypanosoma and Sarcocystis. Another direct route of transmission is the genital route, through coitus. An example of protozoan parasites that is being transmitted via this route is the Trichomonas and the Trypanosoma equiperdum. So these are also known as venereal infections. Another mode of transmission is the congenital route of transmission, and this is exemplified by Toxoplasma gondii, which belongs to the phylum Apicomplexa. So when we say congenital infection occurs when a mother infects a fetus before its birth. Congenital infection occurs via vertical transmission, meaning that the virus is spread from one generation to the next generation. Another mode of direct transmission is via the crop milk, and this is exemplified by the protozoan parasite Trichomonas gallinae. So the pigeons suffering from trichomoniasis shed the parasites through the saliva as well as the crop milk. When we say crop milk, that is a secretion from the lining of the crop of the birds, of the parent bird, that is regurgitated to young birds. For the indirect mode of transmission, we have the vector, through the vector, when we also have through mechanical transmission and biological transmission. So when we say vector transmission, this is a strategy used by protozoan parasites which inhabit the blood or internal tissues within its host. So this strategy involves a hematophagous arthropod. For example, when we say hematophagous, that is a blood-feeding arthropod serving as intermediary between successive vertebrate hosts. So example of human diseases that can be caused by protozoa transmitted by a variety of arthropod vectors include the Trypanosoma gambiense, Trypanosoma brucei, which can cause African sleeping sickness, and this is transmitted by the arthropods, chacha flies, and the Glossina. We also have the Trypanosoma cruzi, which can cause Chagas disease in humans, and this is transmitted by the kissing bugs and the triatoma. We also have the leishmania. Leishmania can be transmitted via the phlebotomus species, and the leishmania can cause leishmaniosis. For the leishmania and the malaria, they are usually considered to be biological, transmitted biologically, while the trypanosomes are transmitted mechanically. So what is the difference between the two? The basic difference between biological transmission and mechanical transmission is that in biological transmission, the agent develops and or propagates within the vector, while in the mechanical transmission, the simple transfer of the agents from one infected host or a contaminated substrate to a susceptible host occurs.